interaction with Pedro. He was taking my evolution course, and he asked me questions after class. He asked me questions as I left the class. He asked me questions as I walked all the way from Weiss back to ABL. He asked me questions all the way back into my office and into my lab. And at the end of that very short conversation, I realized very quickly I needed to know a lot about the student and how I could recruit him to my lab, or at least get him into some lab somewhere. So fast forward a little bit. He went back to Brazil. Our lab was funded to do some wonderful collaborative work, exploring the promise and the pitfalls of environmental DNA on biodiversity surveillance in flowing waters with our collaborators at the University of Notre Dame. And Pedro was brought on in that project and has done a wonderful job. In fact, while Pedro does have an advisor and a committee here, he has also built a great network of folks across the globe, but especially at Notre Dame. And they have actually co-advised him as well. So I want to give a special thanks to our Notre Dame team, and especially Dr. Jennifer Tank, who was truly a co-advisor for Pedro. Pedro was exceptionally productive in his time here. In addition to the 10 papers he published before he even set foot on Rice's campus, he published an additional 10 papers during his time here, 5 of those first authored. They were published in some wonderful journals like Nano Letters, Molecular Ecology Resources, and Environmental Science and Technology. And for those of you who might be from the EEB world, these have a similar impact level as like a PNAS or an ecology letter. So these are great, wonderful publications. Pedro lives in an interdisciplinary world, so he publishes in interdisciplinary journals. In addition to wonderful papers, he gave many talks, including invited talks at national and international meetings. He wrote many grants, including assisting in writing a USDA federally funded large grant to fund some additional ideas, building out of his original work using evolution to surveil the environment. So he's always been interested in how we can be better at surveilling biodiversity and the impacts on biodiversity. In addition to his actual dissertation projects, he has been equally successful in side projects. I will note that he was part of a new species description here on campus, where he described a new gall wasp by the name of Neuroterus valhalla, which is named after the wonderful oak trees outside of Valhalla, where it was originally discovered. That paper was really cool, independent of all the other work that Pedro's done, because it was the first insect description to publish a species description of the morphology and the behavior, along with the annotated genome. And those have not been linked previously in the literature. And so now forever, there'll be a museum specimen somewhere in a genome that will be always linked. And that was a new addition that Pedro added to that world. Pedro is also a great teacher, a great mentor, a great colleague. He won awards like Best EEB Grad Student Talk and Best EEB Grad Student Paper twice. I will stop here. Pedro is now moving on to a wonderful postdoc opportunity, one of many opportunities he had to work with the eDNA Collaborative at the University of Washington. And with that, take it away, Pedro.
Thank you very much, Scott, for the very kind introduction. Thank you, everyone that's here in person or virtually. Appreciate your time. And so today I'll talk to you about a little bit of what I did in the past few years with my thesis, which is the ecology and detection of environmental molecules in aquatic systems. So over billions of years of geological and evolutionary history, Earth's ecosystems have grown to be extremely biodiverse and quite beautiful, if you ask me. But the consequence of that is they are very, very complex and thus very difficult to figure out. Hence, the field of ecology still exists. Over the past more recent history, humans have played a major role in these ecosystems, and we have altered them from everywhere in the planet. While some of humans' impacts are quite obvious and very destructive, others are not so much, and they can have more subtle impacts into the ecosystems. Because these systems are so complex, it's actually very hard for us to pinpoint or predict how we're going to affect these systems before we do. But to understand all of these systems, because they're so complex, the first step is to know which components make up these systems. In the biology world, this is accomplished by what we call biodiversity monitoring. In biodiversity monitoring, you have, when you have an area at a given point in time, you survey which species of organisms you observe in that area. Then you can compile a list or an inventory of all the organisms living in that area. But you may also take a species-specific approach, looking, for example, at endangered species or invasive species of interest. Biodiversity monitoring is useful not just to understand ecosystems at a base level, which is what us ecologists are interested in doing, but also at understanding impact analysis of what we can call to these ecosystems. If we know what they're supposed to look like at baseline levels, then we can understand how we've altered them. This is also an important resource for managers and decision makers, which are trying to preserve these ecosystems for a long term of our history. Traditionally, biodiversity monitoring methods are very diverse, and they change depending on which organism you're interested in analyzing. For example, if you're interested in mammals, you'll be looking into camera traps. If you're interested in aquatic organisms, you'll be doing some traps in streams. However, this has many challenges. You have to train people to do all of these methods, and they can be quite expensive. They're also quite laborious, which can be logistically challenging. And they may be disruptive to these systems, which can be a problem if you're working with endangered or imperiled ecosystems. Finally, they require taxonomic expertise, which means you have to recognize the things you look and the things you collect, so you can come up with an accurate list of which species are present in a given area. What I'm going to work with through my thesis is molecular monitoring, which is an alternative to traditional methods. Molecular monitoring are based on the fact that as organisms are living their lives, they're constantly shedding different types of molecules into the environment, including you right now in your chair. And this includes nucleic acids such as DNA and RNA, but also other molecules such as polypeptides like proteins and metabolites. Molecular monitoring starts with sampling an environmental sample, such as a water sample or an air sample. Then from these samples, we can recover or isolate the molecules that I just told you about. And if those molecules are diagnostic for a given species, we can use the molecule as an evidence that that species was present in that area. The most used molecule by far is environmental DNA. DNA is great for this too, because inherently DNA carries a lot of information, and it's very easily organism diagnostic. Further, the technological landscape we currently live in really favors using DNA. So there's excellent methods for the detection of DNA, such as PCR, which can detect a COVID-1 virus in your blood sample or in your nose sample, or a small quantity of DNA in an environmental sample. And DNA sequencing, which can generate, since the genomic revolution we've lived in the past 2 decades, terabytes and terabytes of data at a very low cost. So there's very good reason to be using DNA. While this may sound like a convoluted, indirect way of figuring out what's where, actually, recent reviews have found that environmental DNA is more sensitive at organism detection than traditional methods. And the probability of detecting something with this method is higher in most cases. It also carries other advantages. For example, it's the same method used across any taxa. Everything is shedding DNA, so we can use the same methods for everyone. It's a non-invasive method. It just uses environmental samples. It can be more easily automated, and it requires little taxonomic expertise. Once your organism is in the database identified, you just compare the sequence you've detected to whatever is in the database, so you have an objective identification. The problem with that method, of course, it's not a silver bullet. It's that it's an indirect method. When you use an indirect method, you lose a lot of information. When you have a fish in your hand or the organism in your hand, you know approximately how many you had. You know exactly when and where it was. And you also learn other information, such as which life stage the organism is in. But when you use an indirect method, such as eDNA, you lose a lot of this information, because that molecule is an evidence that that species was somewhere in that area around the time you sampled it, but you can't be quite pinpoint when and where. And this is what I'm going to solve henceforth. So a way to solve this problem is by understanding the ecology of environmental molecules, which is what happens to these molecules between the time they get released from the organism of interest to the time I sample it in my sample. For example, if I know the shedding rate or the origin of these molecules, I can try to answer how many of my organisms there were. If I know how the molecules get transported in the system, how the DNA can travel in the water, for example, we can tackle where my organism could have been based on where I sampled it. If I know how long these molecules last in the environment, I can tell you when that organism could have been in that area. And then there's other things like the state of the molecule, which will alter all of the other three factors. And that's something we're going to hear about later down in the talk. So my thesis goals are to improve our understanding of the ecology of environmental molecules for a better interpretation of target detections. And also, I will challenge the technological landscape a little bit and expand molecular monitoring beyond just nucleic acids. So over the next 40 minutes or so, I'll go over my chapters, which are 5 chapters. For the first 3, I will talk more about the ecology of environmental DNA. And then we'll go on into the proteins, about the ecology, and then also its detection. So for chapter 1, which was actually published last year on Molecular Ecology Resources, I wanted to tackle environmental DNA particle size and how that influences its detection. To understand this chapter, we first need to understand that eDNA struggles to separate abundance from spatial temporal information. So if you have a sample, if you have a river that flows towards us, like in this system, and you have a single sample of DNA, even if you know everything about DNA, how fast it decays, how it travels, it's actually impossible to know with a single sample if that amount of DNA came from a small quantity of nearby organisms versus a larger quantity of further away organisms, and that DNA you sampled actually used to be a larger quantity that decayed. And this applies for both space and time. Now, I thought of a solution to this problem that uses the ecology of the DNA, specifically the state of the DNA. So environmental DNA has multiple different components, which include intercellular DNA, which are cells shed from the organism, extracellular DNA that's just dissolved in the environment, and others such as particle-absorbed DNA. For simplicity's sake, if we take 2 components, let's say A and B, if component A, for example, starts as more concentrated but decays very fast, whereas component B starts as less abundant but decays slower, then we can use the proportion of those components to inform us how far away our target is. So for example, if I have a small quantity of DNA, but I observe a larger proportion of component A, I can be fairly confident that my source is nearby. Whereas if I observe the same amount of DNA, but I actually see a higher proportion of component B, then that means my source, there's a larger number of them and they were farther away. So let's go back to the same example, again with 1 sample. Instead of just knowing the total eDNA concentration, if I know the proportion of components, then I can use that information to tell me how far my organisms are. So before I tackle this, I need to answer a few basic questions, which is what we're going to do in this chapter. The first one is, do different eDNA components really do decay at different rates? Is this graph real? Will this be consistent across different species? Is this a generalized pattern that can be useful for us? And finally, what's the most appropriate mathematical decay model for these things? So if I'm going to backtrack where my source is, I really need to know how my DNA is degrading. So over the course of my talk, over the course of my chapters, I use DNA from these 2 sources. I use DNA from common carp that we obtained from the zoo, and steelhead trout, which we obtained from the hatchery. We don't really work with the fish. We work with the fishy water, which is just the water that the fish live in, which is really rich in eDNA. That way you don't have to deal with iCooks and that type of thing. So how do we separate the different components? Well, there's not a number of ways to do it, but the way we do it is by sequential filtration. So sequential filtration is when you take a water sample and you pass it through filters of sequentially smaller size. Because components have an intrinsic variation in size, when you sequentially filter, you split those components, and then the size fractions can be used as a proxy for the different components. So my experiment is very simple in principle. I have carp and steelhead fishy water. I put it in some buckets across many reptiles in control setting. I sample it over time, and then I sequentially filter my samples to keep track of what happens to each of my components over time. Then, to recover my eDNA, I use a phenylchloroform extraction. So the DNA is going to get stuck in the filter. When I put phenylchloroform, it dissolves the filter, and I recover all the DNA that's been there. Then to quantify how much DNA of my target there is, I use digital droplet PCR, which is like a PCR, but you split the reaction into about 20,000 droplets using an oil, and then the reaction is going to happen independently into each of those droplets. Then you can account how many of those droplets are positive or negative. You can use a Poisson distribution to quantify how many of your targets are there at very high precision, without much influence from PCR inhibitors and without a reference curve. It's a really neat method. Okay, so what do we see after all? So when you look at the quantity of eDNA components over time, CARP always in blue, still has always in pink. You see here that the quantity of different components oscillates over time. This result is very clear if I look at it with proportions, relative proportions. So when you look here at common CARP, you see that at the beginning, there's a higher predominance of the largest particles, whereas for steelhead, there's a larger proportion of its more equilibrated quantities. So that tells us that eDNA composition changes depending on which species you have. Also, and this was the first time anyone showed that. You see when you look at over time, that we see an increase in the proportion of the intermediate-sized particles and a significant decrease of the largest particles. When you look at steelhead, the pattern is more clear, and you see that the intermediate-sized particles increase in proportion over time. So do different eDNA components decay at different rates? Is this graph true? It seems that it is true. While this is only one bucket experiment, you see that we replicated this over time across all my chapters. And is this consistent across different species? Well, starting composition was different, but they kind of behave the same after a while. So there are some generalized patterns. So the remaining question is, what's the most appropriate decay model for multiple components? And I know what you're all thinking, finally, some math. So as you go see, obviously, I'm not going to have time to go into the details, but I work closely with Daniel Halleck, who is an engineering PhD student at the University of Notre Dame. And through experimentally and mathematically, we're able to note that independent decay model was the one that best described my removal. And that's a model that accounts for each size fraction decay independently at their own rate lambda. So from now on, when I refer to environmental DNA decay rates, I'm referring to lambda. Well, while I got some initial principles for that hypothesis, for chapter 2, I expanded into my findings, and I wanted to see if that also applied to other systems and in flowing water. So my questions here is, will DNA component proportions also change in flowing waters? So is this pattern true for other systems as well? Then I wanted to know what's the mechanism behind the differential removal of the DNA components. What's making them do this? And then I want to know if particle size distribution is affected by environmental factors. In other words, I want to know if this pattern is going to occur independently of where I am. If I'm in like a lowland Texas murky stream versus a beautiful mountain stream, am I going to see the same pattern or is it going to change? So to test this, I will use this recirculating isocosms, which are at the University of Notre Dame. So these recirculating isocosms are kind of like a bathtub, but they have these recirculating paddles here that move the water in a recirculating manner like that. It simulates the dynamics of a moving water system. The advantage with them is we have 24 of them, so we can make experiments in replicas, and also we can put whatever we want inside, so we can see how eDNA behaves under different circumstances. So for my first experiment there, I had some streams where were just empty with no substrate just as control. Then I had the substrate stream where I just added substrate, which is just a physical retention technique. Then for other streams, I added particles that eDNA is likely to interact with in natural systems. I started with fine particulate organic matter, or FPOM, which is just decaying organic matter that's very common in streams and very rich in biotic in bacteria as well. Also added plankton, which is just algae cells that are in suspension in the water column. And I also added clay, which is just inorganic particles that eDNA is certainly going to interact with in the natural systems. Then I completed my array of treatments with titanium dioxide, which is a particle that's well known. It's a chemical contaminant. It's the M and M&Ms, and it's also very well known to interact strongly with DNA. So it was my positive control for absorption. So my experiment looks very similar to the one you've seen before. I take the fishy water, I put it in the stream, I sample it over time, and then I sequentially filter to see what happened with the components over time. My results are going to look like this, and this graph is very confusing. So let me explain to you before I show you everything. So what you're seeing here is the components of eDNA by size and how they change over time in the stream. And you can see that it's reducing in quantity over time. In the inset graph here, what you see is the relative proportion of each component, which is what I mostly look at because it's very hard to see this in this graph. So what do I see across all treatments? So before I get into any details, just by glancing at this graph, you can see that it changes a lot depending on which streams I have, right? The most striking finding here by far is the effect of clay and titanium dioxide. You can see that small eDNA components, 0.2 and 1, are completely gone in the presence of these particles. This is a very strong pattern no one had found before, and it's pointing to me that eDNA is absorbing to clay and other inorganic particles and getting lost in my DNA extraction protocol. I also see here that there is a lot of larger particles in the absence of substrate, whereas when you have substrate, there's not nearly as many. Finally, you see that if I ignore the effect of clay because it was so strong, I see a almost significant decrease of the proportion of larger particles. For steelhead, we see a very similar pattern. So although slightly different, but we see the strong effect of clay and titanium dioxide, and then also the larger particles in absence of substrate. And again, if I ignore the effect of clay and titanium dioxide, we see a significant change in the proportion of components over time. This should sound familiar because we just reproduced chapter 1 findings, except in flowing water. So this pattern was really strong. So I wanted to make a complementary experiment just to see how eDNA was going to behave with all these components without the recirculating water and without the effect of time. So I just took those particles that I talked to you about, FPOM, clay, and so on, took the eDNA, water from each of the streams, mixed them together, and then instantaneously filtered just to see what would happen really quickly. And then I quantified the eDNA into each of those samples. And the results are pretty remarkable. So within seconds, you can see here that those particles have a strong influence of how much eDNA I recover. There's no effect of plankton, but FPOM, clay, and titanium dioxide have strong patterns. And if I look at the particle size distribution of eDNA in the samples, you see exactly what we saw in the streams. We see that relative to control, the 0.2 and the 1 particles are completely absent in those treatments, which is a very strong and fast effect. No one had seen before. So if you look at steelhead, if you just squint at these graphs, you see they're very similar. So eDNA is behaving the same independent of which source it comes from. Well, so answering our questions, do components proportions also change in flowing waters? Yep, we replicated chapter 1 findings, but you see here that the pattern changes depending on my stream composition. Is eDNA, so the other question we have is particle size distribution affected by environmental factors? We saw that it was. For example, we saw that the small particles really do like to bind to inorganic particles. So that's a strong effect. So the remaining question we had is what's the mechanism behind the differential removal of eDNA components? What's causing them to get removed at different rates? So if I look at how fast eDNA is getting removed in each of my treatments, which is the decay rate, like lambda that I showed you in chapter 1, you see here that for total eDNA and for each of the sizes, we see that the only treatment that had a significant effect was FPOM, or organic particles, where eDNA was removed significantly faster. Now, that doesn't tell us very much. So I had to do a more complicated analysis. So in each of my streams, while I was also sampling eDNA, we're also recovering several other ancillary variables. We're serving temperature, pH, water velocity, sampled water for nutrients, we're collecting suspended particles, among other things. So then I analyze this data using a forward stepwise regression. What that does is basically I take each one of my variables and it adds one to the generalized linear model one by one to see which one of them has a significant effect on my explaining my variable. And it does that to avoid overfitting. The result's going to look like this ugly graph you see here, where you see all of the variables here, and then their effect size. If you see a dot, that means that that variable was significant in improving my understanding of the variable. And if you see a red dot, that means that was highly significant. So when I make this analysis with the large eDNAs from both carbon and steelhead, we see that physical parameters are the ones that seem to explain the data best. And this includes water velocity, pH, and suspended solids. That makes sense. Remember, in the absence of substrate, we saw a lot of more of the larger particles. So it seems that something physical is causing them to disappear. When we make the same analysis for the smaller particles, we see a completely different set of variables being more relevant, which tells us that these particles are getting removed by different factors. Specifically, we see that nutrients are very good predictors of how fast eDNA is getting removed. This includes nitrates and dissolved organic carbon. Because we didn't add new nutrients to these streams, this means that there's a variation in the biological activity within the streams that's consuming those nutrients. And this is just a correlation that we see here. So basically, the removal of small particles is associated with biotic components. So answering my question there, it seems that largest particles are removed by physical factors, whereas the smaller ones are more removed by biotic factors. Okay, so far I've talked to you a lot about eDNA particle size, which is the physical size of the particles containing eDNA in them. And that's something we obtain by sequential filtration. Now I'm going to talk about something slightly different, which is the eDNA molecule length. So molecule length is the physical size of the DNA molecule in base pairs that you can recover from the sample. And that can vary within each of the particle sizes. So for chapter 3, I will look at the difference between particle size and molecule length on the ecology of eDNA. This is important because it's well known that DNases, which are the enzymes that eat DNA, will degrade DNA like little scissors, and they'll cut DNA molecules into a smaller size, decreasing their size as they get degraded. This has led other authors to suggest that you can, theoretically, eDNA relative molecule length is going to decrease as eDNA travels away from the source, which means if you detect longer eDNA molecules, that could mean your source is nearby. So the questions I'm going to address here is, will eDNA molecule length really change over space and time? And will the eDNA ecology change for different molecule lengths like it did for particle sizes? So to do this, I went back to my trustworthy mesocosmos, and then we did a simple experiment this time. I had the anti-streams as a control, and then we added substrate for a physical retention of the particles, we added physical factor, and then substrate colonized by biofilm, which adds that biotic component to our experiment. Then my experiment looks very similar to what you've seen before. This time I only used steelhead DNA for simplicity. We added the fish water to the streams, sampled it over time, and then sequentially filtered. But then for analysis, I did something slightly different. I looked at on the eDNA literature, and it helped that there was a recent review, and looked at how big are the eDNA fragments that people are detecting in the environmental samples. And we saw this distribution that really favors small particles. So I designed 2 assays to detect steelhead eDNA that capture about 90% of this variation in the eDNA literature, which are 86 and 387 base pairs. So what do we see when we analyze the data with the different particle sizes? So let's just look at short molecules first. This is the same graph you've seen before, except they're split now. So you can see here that eDNA concentration decreases over time, and then we look at the proportions, we see very similar pattern that we had before. In the absence of substrate, there's more of the larger particles, and we see a significant increase in the proportion of intermediate sized particles. This may sound familiar again, because again, we just reproduced chapter 2 findings. Now, if I look at the results with the longer molecules, it's very much the same. So we'll do that thing again where we squint at the graphs, and you can see that they look remarkably similar from each other, although slightly different. So that's telling me that those components are behaving approximately the same. And there's no significant differences. So if you look at how fast eDNA gets removed, so the removal rate, we see that when we look for total eDNA, the only treatment with a significant effect was biofilm, where eDNA was removed faster. Now, if we do this analysis for different eDNA particle sizes, we see that this effect was way stronger for the smaller particles, the 0.2 particles. And that was where that's the only place where it was also significant. So this makes sense because remember, I just told you that smaller particles seem to be more removed by biotic factors. So the fact biofilm affected more makes sense. And again, we saw that physical retention of larger particles in the absence of substrate. So if you look, and then we also saw that independently of the molecular length, the pattern seems to be the same. All right, so answering my questions at the beginning, will DNA ecology change for different molecular lengths? No, it seems that the molecular lengths, at least within the range we analyze here, behave very similar. And the question we still have is, will DNA molecular length change over space and time? So obviously, the concentration of DNA for both molecular lengths decreases sharply over time. But if I look at their relative proportions over time, we get this very confusing result, where you see that the longest molecules seem to be increasing in proportion over time, which is exactly the opposite of what we expected. Now, if I look at the effect of substrates, it looks the same. But very interestingly, when I look at biofilm, we see the reverse pattern, where now the molecule lengths proportions seem to be diverging instead of converging. That's telling me that biofilm is preferentially or is using more of those longer molecules. Now, if you go back and look at the microbiology literature, you find that this makes very much sense, because as things with biofilm, it's well known that they will use long eDNA molecules and their sticky nature as adhesive for their biofilm structure. In fact, there's been studies that show that if you remove long DNA molecules from systems, biofilm can no longer form. So this is proposing a mechanism that potentially explains this association between eDNA removal and the presence of biotic factors. It seems things are using DNA as building blocks. So if we go back to that first assumption, is it true that longer DNA molecules will mean a nearby source? Not really. We saw long DNA molecules present throughout our analysis. However, it seems that their proportions will change just like particle sizes did. So the proportions may be informative. All right, so now we've talked a lot about eDNA for 30 minutes. We can go over just the general conclusions of what we've learned. So we see that eDNA is composed of different components which vary in size, and that by sequentially filtering, we can obtain a lot of information about the ecology of eDNA. I showed you that different components decay at their own dependent rate, and that those different components seem to be decaying by different mechanisms, whereas large particles seem to be removed by physical factors, and smaller particles seem to be removed by biotic factors. This is good because we may predict how eDNA behaves in different systems. A mountain stream that has more physical parameters will have a faster removal of these, whereas a slower moving, more nutrient-rich area will probably have a faster removal of smaller particles, for example. We also saw that small eDNA components are subject to absorption, and this has a lot of consequences for people trying to capture eDNA in turbid areas. If you're in turbid areas, you should probably use really big pore sizes because you really want to recover the DNA that doesn't do that. We also saw that molecular length doesn't seem to change DNA ecology, at least within the range that we analyze here. But the most important finding is, of course, that eDNA component proportions seem to change over time. And this applies to both particle size and molecule length. And that we can potentially use the different proportion of components to tell us more information about where our source is. But if you remember my introduction, I told you that organisms shed more than just eDNA. In fact, they're constantly releasing all of these molecules, RNA, polypeptides, and metabolites. While DNA carries is very easily the organism diagnostic, other molecules are also the agents driving many of the biological activities. So if I detect an organism based on the presence of a protein, for example, not only does it tell me that organism was present, it can also tell me something about what it was doing in that system. So that potentially gives me more information. With this in mind, in chapter 4, I was looking into environmental proteins by using genetically engineered crops. What? Yes, genetically engineered crops are engineered to produce this protein called Bt toxin. Bt crops, as they're called, have grown rapidly in popularity in the United States since they were approved in 1995, and now represent over 80% of the planted area of most crops. And it reaches similar percentages in other developed countries where this is allowed. BT-Cry is really useful and really smart because when the plant produces this protein, when the pest insect eats the plant tissue, this protein will enter their digestive tract. It will get solubilized and activated, and then it will create pores in their intestines of the insects, killing them inside out. This is really smart because it's killing the pests, but only the pest that eats the crop tissue, which is good for other things living in those ecosystems. And it's also safe for us. We're going to eat those crops later because we can't solubilize and activate those proteins to make them toxic for us. This is really advantageous for a few reasons. It increases overall productivity. More productivity means less area being used for agriculture, which is good for the environment. And also, it reduces the use of broad-range pesticides, which are also damaging. The problem we found is our collaborators have found that these BT-Cry components seem to be escaping from the crops. They persist for a long time in the detritus that's left behind after harvesting. And because these things are very close to ecosystems, such as in stream, like you see here, when these components accidentally get into the system, the cry proteins seem to be leaching and escaping from the environment, to the point that if you go out there and you sample a stream, as you see later, you're almost guaranteed to detect BT-Cry toxins. And this is a potential problem because now these proteins are not only affecting things that are eating the crops, but also other organisms like this hydrocyclic caddisfly that's just living its life inside of the stream. So there's two reasons to be monitoring BT-Cry. The first, of course, is system-specific, which is to monitor the impact of GMO crops. But I also wanted to use this as a model to understand how wheat proteins are going to behave in the environment. So there's two reasons here. So our questions are, how long will wheat proteins last in the water column? And what are the determining factors for the fate of BT-Cry wheat proteins? And if you want to take a guess of how I did that, yes, I used recirculating mesocosms again. Except this was my first ever recirculating mesocosm experiment, and it was done in the original recirculating mesocosms at the Carey Institute of Ecosystem Studies in Millbrook, New York. They are just like the ones I showed you before, except they're twice the size. So for those, I also had different stream treatments to see what would happen with the protein under different conditions. I had the empty stream as a control. I had some leachate, kind of like a tea. And then I have a trustworthy FPOM and different types of substrates, which were colonized by biofilm. And finally, a mixed stream, which is just everything put together, which is what the protein is actually going to experience in the wild. My experiment then looks very similar to what I've seen before, but instead of using fishy water, this time I use corn tea. So I take the dry corn tissue with which the cry protein persists inside. Then I make this corn leachate. Then I take a sample of here to quantify what's the concentration of my protein in this leachate, and I add it into each of my streams. Then we sample the water, and I filter it with, again, but instead of worrying about the DNA that got stuck in the filter, this time I'm concerned with the protein that went right through. I freeze that sample, and then I concentrate it using an Emicorn filter because the protein is not concentrated enough to be detected. And then I analyze it with ELISA. So what we see here for the concentration of wheat proteins over time, you see that we had a starting concentration of about 175, but the treatments have a dramatic effect of how much protein was detected within the first hour. You see here that there is strong variation in how much protein was detected. And that by 13 hours or so, all the treatments seem to equalize. And we still had protein detected in all treatments by 48 hours, which is telling me here, what this is telling me here is that there seems to be a biphasic decay. So if you look at just FPOM here for clarity, you see that the starting concentration is here. There's a very sharp, rapid removal within the first hour, followed by a slower second phase of removal. When we quantified the dissolved organic matter in these streams, we noted that would get removed at the same rate as FPOM, as BT-Cry in this slower phase. I know you're thinking now, yeah, we've seen this rapid removal with FPOM before. That's absorption. That's what happened with eDNA. No, because that also happens in the absence of FPOM, such as when I have leaf treatments. Here, I also see the rapid followed by the slower removal. So it's something else that's happening here. Now, I also collected a lot of ancillary variables in these streams. And when I look at which one best predicts how fast my E proteins get removed here, I see that what the best predictor for how many cry protein was remaining after 1 hour was ecosystem respiration. Ecosystem respiration is a measure of how much oxygen is being used in that stream. So that means more heterotrophic organisms. If you're a gene bro, you'll know that proteins are a really good protein source. So I think that what's happening here is exactly that. It seems that these heterotrophic organisms in the streams are releasing proteases in the water column, which rapidly remove the protein from the environment. Now, interestingly, I also saw that this pattern persisted in the absence of biotic components. So I did a very quick experiment in the lab where I took the same water from the streams and I measured pH from 4 to 10, which is a range we see in the streams. And then I put cry in it just to see how much cry would persist after 1 hour. And the results were pretty clear. We saw a very strong effect of pH on the protein. And again, this is a laboratory vial, just a very small vial. And I only recovered between 60 and 30% of my protein after 1 hour. What's probably happening here is that the protein is quickly denaturating. If you ever work with protein, this is no surprises. They're really finicky. If you put them in the wrong media, they will denaturate really fast. So it seems that this is what is happening with E proteins. So answering my questions here, how long will E proteins last in the water column? So we see that they are rapidly removed at first, so within the first hour, and they persist at low rates and get removed at the same rate as organic matter. Because I never see it being completely gone, I can't quite tell you how long it will be until they're completely gone. Now, what are the determining factors for the fate of cry proteins? Well, I just showed you that biotic activity and water pH are both strong predictors of how fast eDNA is getting removed. So it seems that those proteins are quickly being used as nutrients or just denaturating, which is good because they lose their function. This is good-ish functions for the good news for the safety of BT crops. These crops are growing very near to this ecosystem. So if those proteins are constantly getting leached in the system, they are quickly decaying, which is good. So if you keep track of one cohort of molecules, you see that that decays very fast. But because there's a constant source, that makes BT a pseudo-persistent contaminant, which means it's always present, but its impact is limited to a close range, or where the source is. And I think this will likely apply to other E proteins as well. Proteins are very different, but they all seem to denaturate and be used as nutrient source. So I think that if you are out there detecting E proteins, their range is going to be quite short. Well, I told you, there's very good reason people are not using proteins and are actually using DNA. And that has to do with our technological landscape. There's no method for protein sequencing that's very good, and DNA and protein detection is accomplished by ELISA, like I did. So for chapter 5, I wanted to challenge this technological landscape a little bit and develop a new method to detect E proteins. And this was done, and I was lucky to do that with the work from Dr. Carol Tanner and Dr. Stephen Bergeros from Notre Dame, who developed the light transmission spectroscopy, or LTS. So in LTS, you take a laser and you shoot it to a reference and your sample, and then a detector will tell you how light got reflected by your sample, which is predicted by the theory of scattering, which depends on how many particles you have, their material, and the size of the particles. So LTS can tell you the size of the particles within each of your samples. The major advantage with LTS is it removes the effect of background because it's reading both a reference and your real sample. So when you have your sample, and you can add that reference, which is your environmental sample, and it gives you a clean extinction pattern, which is ideal for the complex environmental samples that we deal with. Finally, the equipment itself is field deployable. It's about this big, and it can be theoretically powered by a car battery. So it can be done in the field and very fast. Now you're probably thinking, what does that have to do with anything? Well, if you're smart about this, you can actually detect eDNA using this device, which Scott has done before. So they have developed this nanoparticle, which is coded by a probe that detects your organism of interest. If you put this in the LTS, it'll show you a single peak, which tells you, yeah, I see your particle here. Now, when your organism is present in your sample, in this case, zebra mussels, they saw that those probes would recognize the eDNA of those organisms in the sample. And then when you put that in the LTS, now the LTS will show you two peaks, which represent this and this. So this basically, the second peak can be used as a diagnostic for the presence of your organism of interest. What I did is I designed a size shifting assay to detect E proteins. So my assay starts with gold nanospheres, which can be manufactured to very precise sizes. Then nanoparts functionalize them with antibodies that are anti-my bt-cry protein. What that does is when my protein is present, my gold nanoparticles are going to recognize the E protein, and then they clump around the protein, forming this larger particle that can be potentially detected by LTS. So when I take my gold nanoparticle and I put in the LTS, this is what it spits me when there's no BT present. It gives me two peaks, which I think has to do with which angle the gold nanoparticle was at when the laser passed through. So it's that sensitive. Now, if you put the bt-cry proteins, you see that the peak, we have a new peak appearing, which is way larger and represents that clump that we see forming with the protein and the gold nanoparticles. And this peak only appears in presence of bt-cry and can be used as a diagnostic for its presence. If I repeat this over and over and over again with different concentrations of bt-cry, I get this curve of the probability of detection based on the concentration of cry. And I see that for 95% confident detection, you need about 1.5 nanograms of proteins per mil. If I compare that with the ELISA LOD, you see that the method is actually not quite as sensitive as ELISA, which is the current gold standard. But remember, ELISA has been around for dozens of years. People have been optimizing it ever since. And this is just the first time anyone has tried LTS. Further, LTS is still deployable and it's way faster. So maybe we have something in our hands here. Then I prove the method by collecting samples in streams. Like I told you before, if you go out and just sample them, you reliably detect bt-cry proteins in the samples. So I ran these samples through the LTS, and I found that LTS was able to detect cry proteins in these samples most of the time. So our technological landscape still favors nucleic acids, and it's not like one method is going to change that. But I can argue here that protein detection in environmental samples is quite possible, and it may be useful in several cases. So with that, I have a lot of people to thank, including everyone that just listened for me for 43 minutes. And then acknowledge all of my co-authors. I collaborate with 16 different scientists over this project. Without each one of them, this would not have been possible. I also need to thank the Egan Lab, other RISE staff that have helped me along the way, the committee members for being with me for the past 6 years and being very encouraging. Thank you. I also have to thank other collaborators. There are other things I've done, like Scott told you, that didn't make it into my thesis, like the Valhalla wasp that's in my tie here. And I need to thank all of those people as well. And other people that made this possible, including, of course, the funding agencies that believed in us and gave us money. So I also, on a more personal note, I have many, many people to thank. Everyone that I've met here at RISE over the past years have kind of helped me rebuild that support network that I left behind in Brazil. So I'm very grateful for everyone here. Please don't be sad if your picture is not here. It turns out, and when I was collecting pictures to put in the slide, I found out that I just don't take enough pictures. And so I have many people to thank. People at the Beer Bike, the Brazilian Student Association, my local family that's right here, my lab, my roommates, the dogs I took care of, many people, and I have to thank my cousin David too. So he gave me a scholarship to study English. Without him, I wouldn't be here talking to you right now. I also have to thank my family who supported me and believed that I could make a career in biology. And yeah, and the most special thanks, of course, goes to Lisa, who moved across continents to live with me. And there's really no bigger proof of love. So I love you all. Thank you very much. And I'm happy to take any questions.
Okay, I have two questions.
Yeah.
The first one, what is the source of the biofilm that you use it for research?
Mhm.
And the second one, I forgot the name, but the experiment that is simulating the stream.
Yeah.
Have you tried different speeds? Because I was wondering how the speed of the water would affect your final sample as well.
Yeah, congratulations, you've unlocked the next slide. So so you asked what's the source of the particles? So we collect the particles in natural systems, but the biofilm, we actually put the rocks to be colonized at another experimental stream that exists in Notre Dame. It's called the NDB. Then we take those rocks that's been sitting there for 6 months and then we put them in the streams. But your second question was if the velocity changes? Yes. So the paddle wheel in those streams, you can actually modulate them to change the speed. The speed that we did is representative of what you see in an actual stream. If you can put it way faster, it starts sloshing around, so it doesn't quite work. But you can modulate it, and there's been experiments that have done that, but I just didn't do that now. Yeah. Yeah. Yeah.
Okay, first of all, congratulations. Huge amount of work, beautifully presented, very charismatically presented. If the slow biology thing doesn't work, well, I'm pretty sure you will. Have you considered an alternative career as a stand-up comedian?
No.
So that's not my actual question.
Okay.
You can say that, but my actual question is actually a very big picture question.
Yeah.
So when Scott was introducing, he said that you visualize to work on the promises and pitfalls of eDNA. I was wondering if after 6 years with this, have you become more excited about the promises, or has you become more concerned about the pitfalls of eDNA?
Definitely more excited about the possibilities because so when I first encountered eDNA, it was 2012-ish. It was a paper by a Japanese group. And then also Dr. Kelly, which I'll be joining in the postdoc, also did a paper around that year, which showed eDNA. And since then, I've been like, I want to work with this. And but since 2012, so many papers have come out, which in an experiential manner, and it seems that everything people try to answer with eDNA, it just works, which is crazy. And I hope to continue doing that for the future for sure. No. What is the question?
Here, I can read it.
Yeah.
You want to read it?
No, go ahead.
Okay, all right. So question from Hannah.
Mhm.
How much do you think the potential similarities and differences in microhabitat use by the trout or the steelhead fish could affect your results?
Yeah, that's a good question. So we're not sampling the samples in a real system. We're collecting the water from a zoo or a moat or like a hatchery that hatches them. Those are different conditions, and we definitely think that that could affect the composition of eDNA, right? Now, your question about the microhabitat, there's been other studies that have shown that, for example, if you sample a lake at different depths, the fish or the organisms you detect at the deeper areas are organisms that are known to inhabit the deeper areas of lakes, versus if you sample the shore, you detect organisms that are known to hang out on the shore. So eDNA signal can definitely predict microhabitat use, but it just doesn't apply to our work here. Thank you. Yes.
So, like in the first two chapters, you talked a lot about like the different factors that can change, like the fragment size and different rates and sort of things. And there was a lot of context dependency.
Yes.
In general ecology, we get excited about context dependency, except this is a method.
Yes.
Where we don't want as much context dependency. So thinking about, like my community, we have a bunch of different species and each have a species-specific pattern.
Mm-hmm.
We have a lot of organic matter in play and a lot of this stuff, which is like everywhere. So what do you think is like how useful and like how concerned are you like extrapolating these things for like community analysis where like you don't calibrate everything for specific species?
Yeah, that's a great question. So we do see that this pattern here, like I showed in this graph, seems to be species specific because the starting proportion of components is different, right? However, I did show you that the eDNA seems to behave the same independently of which source it comes from. So I think that while the starting proportion may be different by species, the general pattern of decay is going to be the same independent of who the DNA belongs to. So if you are to apply this method here, you should probably know beforehand what's your starting composition of your target organism. And you should have an assay of how the eDNA is going to remove in your specific system before this can be applied. But eDNA just works irregardless of the system, right? It's the presence absence is going to work. It's just that if we want to get more information, then we'll need more information from the system. But yeah. Yes.
Use eDNA and proteins. I was curious about metabolites.
Yes, so I didn't use metabolites here. This was something I was exploring with a couple postdoc applications with Dr. Brian Sebio at UT. So they can do metabolomics with environmental sample. And I always thought that was really neat. You could also think it from a spatial perspective. Like we see oxygen in our atmosphere, that's a metabolite, right? And the presence of oxygen means something is present, right? So people can do that over space as well. So, but I haven't explored that yet, but I think that's something that's gonna develop as those methods develop too. Yeah. Yes. Yes. So right now, DNA is obviously better, right? If you want to detect an organism. But like I said, the protein carries function as well. So let's say I detect an enzyme that digests oil or something like that in the environment. That's not only telling me that that organism that digests oil is present, it's also telling me that it's needing to digest oil, right? Because it's producing that enzyme. So it carries more information. Now, a lot of what you can accomplish protein, you can also do with RNA, right? So RNA is also produced conditionally, and RNA can benefit of PCR and DNA sequencing technologies. The problem is when I did this work, we pretty much assumed that there would be no environmental RNA, right? Because RNA is very finicky. Like everyone that works with RNA says, if you breathe wrong, it degrades, right? So like there's no way there's going to be RNA in the environment. But since then, it turns out you can detect RNA in the environment. So I think that a lot of the applications that would otherwise be used by proteins can also be used by RNA, but I'm sure there's other applications out there I just can't think of that you use proteins. Yeah.
Were you sequencing the DNA with your analyzing, or was it just cells?
Yes, no, we don't sequence the DNA in this analysis. We just use a primer that's species specific. So we try that primer with other species that are close, and we design the primer so it only amplifies my target. So the primer becomes a diagnostic. It's kind of like PCR for COVID works. The primer only amplifies COVID, although your DNA is also in the sample, it only detects the DNA from COVID. So kind of the same idea. Yeah.
All right, there's just one more comment from online. It's been repeated. It says parabens.
Oh,
I know. All right, with that, we're going to end the public part of the defense. So one more round of applause for Dr.